Okay. So today we're doing the first part of two chapters worth of innate immunity. But one of the tougher concepts is, of course, the complement system, which we'll cover today. So hopefully by the end we can get this straight in our minds. Okay. Right. So the response to infection typically occurs in 3 phases. Hold on.
Yeah, like battery extras. Did you try reconnecting it? Thank you.
This one?
Nothing. Mouse.
How do you do that?
On the back, on the bottom.
On the bottom,
It's a red dot or a yellow white dot. The white dot on the top.
Wait.
Right above. Right. Thanks. Where the little indentation is at the top. Yes, there. Your finger was on it. I don't have to push in there.
Oh, it's someone just need a paper clip?
Okay. Right, so the initial infection occurs, the response to the initial infection occurs in 3 phases. So the earliest are the things that were discussed today. So these parts of innate immunity that don't require any recognition whatsoever of the invader happens basically immediately, and many times can prevent infection or remove the infectious agent. Followed by what I think we're going to cover next week are some early induced responses that are still part of the innate immune response, meaning there's no DNA rearrangement, but it still requires some recognition of the invader to specialize the response. And finally, the adaptive immune system, which we'll learn about later, kicks in, which really is a very effective means to remove an infectious agent and, of course, works the vast majority of the time. So not only does this happen in phases, but it happens with a variety of different mechanisms. And it is necessary because, of course, there are many, many pathogens. And this figure 2 from the second chapter lists some that are pathogenic to humans. And you can see that the way that we get infected can vary. The disease that results can vary. The mechanisms that are utilized by the pathogen can vary. So we have to have a lot of different mechanisms to fight all these different types of things effectively. So some of the various basics is that kind of the first way we sort some of these pathogens is by looking at them as either extracellular or intracellular bacteria. So extracellular microbes include viruses, bacteria, protozoan, fungi, or worms. So pretty much anything initially can be extracellular, but then of course things like viruses and protozoans can go on and infect cells and sit away from a lot of the mechanisms of the immune system. So we have to have specialized mechanisms in which we turn on parts within the cell to actually go ahead and kill these things that have exploited the cell in this way. So microbes in general we think of as viruses, bacteria, and fungi are things studied by microbiologists. And then protozoans and worms or helminths we break down into parasites and are typically studied by parasitologists. But our immune system, of course, has to fight all these different things. So some of the different ways that pathogens can lead to damage to the human body or tissues So at times there are some extracellular bacteria that will secrete a toxin that can be directly toxic to different cells in the body. Here are some examples of agents that will do that. And then there are things that we would call endotoxin that aren't secreted but are still cause a toxic response by direct contact with part of the host or the human body. And then, of course, they can have a direct cytopathic effect that the infectious agent itself can be directly toxic to cells. And then there are some indirect mechanisms by which pathogens will cause injury to the body. And they can lead to immune complexes. So, of course, our immune system is kind of participating in the damage in this way that will lead to kidney disease and vascular deposits. And then there can be parts of the pathogen that are so very similar to something on the host that we accidentally and inadvertently create anti-host antibody that can result in autoimmune syndrome. And then, of course, when things invade cells, one of the ways we deal with it is actually to upregulate T cells and NK cells that will directly kill these cells, so you can actually get quite a bit of toxicity just trying to control intracellular infection. Right, so all these different mechanisms, so these early stages to these late stages, happen as the pathogens kind of increase their penetrance into the body. So some of the earliest parts of the innate immune system is, of course, just direct barrier defenses. But the way some pathogens get through this is they can actually hear and sneak through the epithelium, particularly in the lungs. And then sometimes there is a wound, so penetrance is easy by these pathogens, and they can get into kind of these the underlying tissues very easily, where there are things like macrophages and dendritic cells that have to kick in and recognize the pathogen and upregulate. And then, of course, what we very much worry about is if these pathogens get past this and they actually get into the bloodstream, and they can cause sepsis. But hopefully by then, things like the dendritic cell will have started the adaptive immune response, and this adaptive immune response will secrete very powerful tools like antibodies and T cells that can get rid of this infection and clear it before it actually enters the bloodstream. Okay, so getting into the different things that the immune system has in order to be able to fight the pathogens at the various phases. So, right, so some of the very earliest and most basic is mechanical, like I mentioned before. So these epithelial tight junctions are located pretty much anywhere, like the skin, gut, lungs, eyes, nose, oral cavity, where you would anticipate that pathogens could get through. There are other direct mechanical means that we see, of course, clinically a lot, like mucus. You make a lot of mucus that can capture the pathogens and prevent them from being able to bind to anything to get further into the body. And then that mucus can actually be moved by peristalsis in the gut. It can be moved by cilia in the lungs, and we know this is very effective because if there's a problem with this, like ciliary dyskinesia, then you get many pulmonary infections. And then things like tears and nasal cilia can also expel things out directly. Which basically comes in the same place that these mechanical barriers are chemical barriers. They come hand in hand, and we'll talk about these just in a little bit more detail. But some of them are very simple and maybe not directly cytotoxic to pathogens, but things like fatty acid layers, a low pH, which would cause direct death of the pathogen, pulmonary surfactants, enzymes, and then some basically antimicrobial enzymes and proteins that are made by the body, like alpha and beta defensins, cathelicidins, reg-3, histatins, all things that would be produced by the body to actually be natural antibiotics, I think of them. But if none of these have worked, excuse me, at these same epithelial junctions tends to be our normal flora. And this is really something that is very new to us in the way we think. But along the GI tract, and of course all throughout the skin, we have a microbiota that we live in a symbiotic relationship with that are not pathogenic to us, and they actually deter pathogenic microbes from coming along. So what we are concerned that when we kind of take a nuclear bomb approach to getting rid of pathogens, we're also getting rid of our normal microbiota and getting rid of this entire line of defense. Of course, when we take antibiotics, it's going to kill a lot of our gut microbiota. When we take bleach baths for atopic dermatitis, while it gets rid of staph and pathogens, well, probably it's getting rid of our normal flora too that's going to help us fight off some of these pathogenic infections. So kind of modern immunology is now taking a look and trying to see if we can come up with techniques to get rid of pathogens or help our immune system get rid of pathogens and nurture our normal symbiotic microbiota.
Is the microbiota resistant to the beta defenses, or is that something we need to keep them in check?
Yeah, so that's a good question. So they live alongside now. There are things like the cathelicidins that are only upregulated during the times of infection. So at the same time, are they going to kill some of the normal microbiota? Well, that may be true, but it's probably going to be local, and it's probably going to be able to reproduce and recover. It's possible that they've evolved to having some, the reason they can live symbiotically with us is because they are resistant in some way to defenses, things that are, constitutively expressed. I just don't know what those mechanisms are. My guess is it's a combination depending on which of these antibiotics, antimicrobial things you're talking about. Okay, so the first one I talk about in this chapter is something called lysozyme, which is an antimicrobial enzyme. It works by breaking a chemical bond in peptidoglycans. It's secreted in tears and saliva and produced by Paneth cells and phagocytes. So it goes into greater detail in figure 2.7 and shows that the lysozyme actually digests the wall of both gram-positive and gram-negative bacteria, but is more effective against gram-positive bacteria because the peptidoglycan is more accessible. So here the peptidoglycan is shown by these purple circles and what are these, like hexagons, these kind of teal hexagons. The lysozyme actually targets between these two components, which are called, and I'll not know this, N-acetylglucosamine, which are the turquoise hexagons, and the N-acetylmuronic acid, which are the purple circles. So the lysozyme actually goes in between these and is able to digest it, exposing the lipid bilayer, making that bacteria more vulnerable to cell death. So you see in these gram-positive, these components are readily available, so the lysozyme works very well. They're somewhat protected by things like lipopolysaccharide, LPS, which we also refer to as endotoxin frequently. These layers protect the peptidoglycan, so it's harder for the lysozyme to work in this case. But of course, there's other antimicrobial peptides like defensins, cathelicidins, and histatins, which are generated by proteolysis of propeptides, and this is actually a major concept to hold on to in this chapter, is a lot of these antimicrobial defenses, even complement, start as a propeptide and then undergoes proteolysis. So part of that that protein is cleaved and makes this active component. So one of them is called asdefensin. It's stored in the primary granules of neutrophils. I probably remember that. There's one called cryptin that is secreted by Paneth cells in the gut, but it's all alpha defensins. There's beta defensins that are not constitutively expressed, but is produced in response to microbes. It's made by the epithelial of the gut, skin, tongue, respiratory, and urogenital tracts. this parenthesis is misplaced. Figure 2.8 actually goes into a little bit more detail about the beta defensins. So defensins are an empathic peptides that can disrupt the cell membrane of the microbe. So does anyone know what empathic refers to? So ampha, right, so it's usually people, right, ampha is two, or it has two mechanisms, right? An amphibian can live inside or outside water. Well, this is very similar. So empathic peptides have two components. So a two-part that is hydrophobic and part that is hydrophilic. So the hydrophilic part, almost, I guess not exactly instinct, but kind of instinctively it wants to. It's uncontrollable forces that's trying to drive it in away from exposure to water. So it's going to try and get into this, this lipid bilayer. But then it's still going to have this hydrophilic part that's charged and can be outside. So what it can do is it can insert itself in and then actually disrupt this membrane and create a pore and lead it to cell death. We'll come across this term again, empathic, when we talk about hydrophobic and hydrophilic. This is something that's actually important. This is one of the main properties when they're trying to determine how proteins fold, and they're actually, we know that some amino acids are hydrophobic and some are more hydrophilic depending on the charge. So when we're trying to predict how a protein would fold, so if you have a mutation, now that we can sequence, right, we can sequence the whole genome, or we can sequence the gene, and then we can look at the mutation and we can see which amino acid substitution occurs, and then we can try and predict how that protein has folded based on the amphipathic nature of peptides. So we can see what part wants to be on the inside of the protein away from water, and we can look and see what wants to be on the outside close to water. And there are very sophisticated computer software programs that we can use to try and determine, well, okay, we see this mutation, but we all have millions of mutations. This one has led to an amino acid substitution. Well, would that amino acid substitution make any difference as this protein is folded? And we can actually look at it and say, okay, well, this is significantly different. This is a nice candidate to be disease-causing. Okay, moving on. So some other antimicrobial peptides included the cathelicidins. They're found in secondary granules of neutrophils, and they're activated after the neutrophil encounters a microbe. It's activated by something called neutrophil elastase. So I just want to point out that we've heard that alpha defensins, is that true? Beta defensins? Alpha defensins are in the primary granules of neutrophils, and cathelicidins are in the secondary granules of neutrophils. I think nice facts to remember. The statins are active against fungi and produced by the parotid, sublingual, and submandibular glands. And then lectins. A lectin is anything that will bind to a carbohydrate. And the example they give here is REG3, which is secreted by the Paneth cells and secreted into the gut, where it can also bind to acetylglycan and disrupt the cell wall of a microbe. So I'd kind of commit all of these, the defensins, the cathelicidins, the statins, lectins, lysozymes, into memory as your natural antibiotics, things that are there ready to try and kill the microbes. All right. So this is a slide on page 47, figure 2.9, that actually just reinforces that concept that everything starts as these propeptides, and then part of it is cut, or the protein is lysed to release an active component in then this pro region. And once it's activated, then it is effective in defense. And depending, and this is kind of implied in what we've talked about so far, but in figure 2.1, we see that this varies depending on where your body would actually encounter a microbe. So at the epidermis, of course, there are very tight layers that prevent an invader from crossing, and then there are laminar bodies that are going to secrete some of these antimicrobial peptides. Of course, in the respiratory tract, you have things like goblet cells that will produce mucus, in addition to secretion of antimicrobial products in the cilia, which will beat this mucus out. And then, of course, the gut epithelium, there's mucus, there's the panocells that are making things like the alpha defensins and reg3. You have your commensal microbiota that is helping prevent infection by pathogen, and then, of course, you have peristalsis that's going to beat all of this out. Okay, everyone kind of understands the basics of that first layer defense, right? So it's barrier, chemical, mechanical, that's going to beat out microbe invaders, microbial invaders. The rest of the time, we're going to talk about complement, and I think complement gets a little bit intimidating, but hopefully we can break it down, try and remember it a little bit better. Right, so the complement the complement system is just a collection of proteins in body fluids that work together to destroy microbes. All these different proteins have different jobs, and they're kind of outlined here in figure 211, which is on page 50. Right, so each of these, just like the propeptides of the antimicrobial substances we learned about before, they start as a zymogen, and then they get cleaved and activated. The zymogen is the complement before it's cleaved and activated. Okay. Now here are all the different, probably 30 or more different complement proteins. The ones we talk about most are the, well, like the things that actually lead to the Macatech complex, right? So Janeway actually emphasizes this less than Abbas ever did, but one mechanism by which complement works is it makes these membrane attack proteins into a pore, and it punches lots of pores in the hole, so it can be directly cytotoxic to cells. But then at the same time, you have these parts of complement that will directly bind to a microbe and opsonize it, and these will have smaller pieces that, you know, it's easy to think of as getting discarded, but they actually mediate inflammation. And then there's a whole host of regulators that keep all of this in check. Now, complement, because all of these parts and things are very well defined, I think is a fantastic target for lots of boards questions. So I would spend lots of time memorizing what all of these do and what diseases you would get if you have a problem in each of these parts. But that's getting ahead of ourselves. Okay, so let's study the kind of the cascade that occurs to fight microbes. So the cascade can start in 3 ways. So one called classical pathway, which we call the classical pathway because it was discovered first. One called the alternative pathway that was discovered second and thought to be the alternative to the classical pathway, and the one that was discovered more recently called the lectin pathway. So does it matter how they start? The goal of all of them is to create a C3 convertase. C3 convertase, its sole job is to cleave C3 into 2 components, C3A and C3B. Okay, so once this happens, the different mechanisms can occur. So C3A can go on and recruit different cells and start inflammation. C3B can tag onto the outside of a microbe and opsonize it for a phagocytosis to come along and engulf it and destroy it. Or it can go on, C3B can go on to make a C5 convertase by the addition of one more protein, and that C5 convertase will then cleave C5 into C5b, which starts the placement of the terminal complement, C5b, 6, 7, 8, 9, and a poly C9, which creates a pore in the side, and with lots of pores, you can actually directly kill the cell because you're disrupting the cell membrane. So in the innate immune system, actually a lot of what's happening is either disrupting the cell membrane leading to death, right, or making it more desirable for other parts of the immune system like the phagocytes to engulf and destroy. All right. So the first one they talk about, right, is the lectin pathway. So how does this start? Well, there's something called mannose-binding lectin that will bind to a mannose that is, drawn like this, that comes along with two associated proteins, MASP1 and MASP2. So these three things together can generate a C3 convertase. Similarly to MBL, there's a cyclin that is also closely associated with MASP1 and MASP2. And this trimer can make a C3 convertase. Okay. MBL, of course we have to remember, because MBL is associated with a deficiency, so a deficiency in MBL can actually lead to a type of immune deficiency. Not one that we can treat here in the States, but one to be aware of. Right, so the target of a lectin, remember, is a carbohydrate. So mannose-binding lectin is a lectin that's going to bind to the carbohydrate that is mannose, right? So a lectin is binding to mannose. So mannose is found on things like yeast, but it's covered up in vertebrates with sialicylic acid because the terminal parts of certain residues or glycoproteins, sialicylic acid in vertebrates. So this is good because we have mannose-binding lectin and we don't want this starting on our own cells. This is one way in which the host prevents itself from being consumed by complement. So the mannose-binding lectin is targeting carbohydrates that are available to be bound to in microbes, but not on vertebral cells. Okay. So the mannose-binding lectin or cyclin in conjunction with the MASP1 and MASP2 are going to bind to the microbe by targeting a carbohydrate. And when it binds to this microbe, it is going to have the capability to cleave both C4 and C2. So it's going to cleave C4 into C4b and C4a. By convention, the A, the vast majority of the time, is going to be the small component, and B is going to be a large component. So C4b is going to attach itself to the outside of a wall, and then C2 is going to bind to it. And C4b, it's really, the way I think of it is 2b. For some reason, C2 is named differently, right? So sorry, it's really confusing the way. I'm actually going to show the boss way they explain this too. Needless to say, C4 and C2 are cleaved by this MBL complex. And these two things together make up a C3 convertase, which is the first major step in complement. So C3 convertase then can come along and cleave lots of C3 into C3b and C3a. So remember, the C3bs and C3as can have lots of different functions. Okay, so, and we're going to go over those in a minute. But once this is made, it can generate lots of C3b. Now, C3b can attach to this, and you have C4b to a C3b, which is now a C5 convertase, which can then cleave lots of C5. All right, now don't let yourself get too upset. Now we're going to look at it a different way. Because it actually looks very similar if you go through the the classical pathway. So the classical pathway requires antibody. So IgM particularly is very, very good at fixing complement and starting this pathway. IgG will also do it. IgG3 is the best, then 1, then 2. So it's IgM, then IgG3, 1, 2. All very good at starting this classical pathway. And the classical pathway starts with C1. Now C1 is made of 3 components that are extremely reminiscent in structure to that MBL and NAF. So you have the C1q, which is analogous to the cyclin or the MBL, and then you have C1s and C1r that are like the NAF. Okay, so when the C1q binds to your antibody, C1s and C1r become activated and do the exact same thing as those NAFs, and they are going to cleave C4 and C2. Now this still has mask, but pretend this doesn't say mask, and this is actually just C1, right? So it's bound, so these are activated, so it's going to start cleaving C4 and C2 in order to make C3 convertase. And once you have C3 convertase, it's going to convert a lot of C3s to the active form, and then a C3 can then bind, and now you have a C5 convertase. So the lectin pathway and the classic pathway are very, very similar. It's just different proteins that get it started. It's either MAS, MBL, or cyclin and MAS, or it's C1 complex. Okay. But there is a third way this can happen, the alternative pathway. And what happens with this is the C3 is just spontaneously cleaved. And it remains cleaved very, very briefly. But if it is close enough to a vulnerable surface, which would be a microbial surface, it can bind, and then bind to something called factor B, right? So another part of the complement that's floating around. And then factor D comes along and cleaves factor B, so now you have C3b-BB, which is a C3 convertase. So now it can cleave lots of C3s. Okay? And then this cascade can perpetuate. So any way that you've made C3 convertase, you're making C3b. And if that C3b can connect to a cell surface, well, then B can come along, perpetuate, and you can get more C3 convertases. And, of course, if you get another C3b to bind to this complex, C3b, BB, 3b, you have a C5 convertase. So in this case, the C3 and C5 convertase are very similar but different than if it's started by the lectin or classical pathway. But there are 2 main goals to keep in mind. So there is going to be a protein that is going to cleave a couple proteins that will make a C3 convertase, which will cleave C3, which will connect to that C3 convertase and switch into a C5 convertase. All right? I know this is so confusing. Once you have a, let's see, I can actually kind of cover this. Once you have a C5 convertase, you're going to start cleaving bunches of C5s. And then C5b can bind to C6, C7, C8, insert itself into the wall. Poly-C9, so it's going to attract lots of C9 proteins. All these proteins are just floating around, right, in the same liquid that, you know, all these complement proteins are together. So they're easily recruited. Once it starts, it cascades very quickly. And this is your MAT complex. This is going to punch holes in the surface. The more C5bs you have, the more you can insert these holes and just start punching holes in the cell surface. But remember, all these things are kind of perpetuating each other. So if you have C5 convertase, you're going to be making lots of C5s, so you're punching lots of holes. If you have a C3 convertase, you're going to be making lots of C5 convertases, and you're going to be punching lots of holes. This is one way that all comes together and works. Another way it can work is that even if you're not punching holes in the cell surface, all these C3Bs are still going to bind to the outside of a microbe, and then it can use complement receptors. So a receptor on the outside of a phagocyte can actually bind to this coated or opsonized bacterium, and it can cause it to engulf. Similarly, things like C5A, so these small kind of byproducts, which we also call anaphylotoxins, can bind to its own receptor and make the phagocyte more active. And also, these C5As can recruit, your immune cells to the site of infection. So you're making C3 convertase in order to make C3B, so it can either opsonize or punch holes, and then it's also going to make C3A.
Attention, if you are the driver of a gray Toyota Corolla, Kansas plates 577ESX, you are illegally parked in handicapped parking and need to move your vehicle. Thank you.
Or you can make C3A, which has, by various mechanisms, can lead to migration of macrophages, neutrophils, lymphocytes to the site of infection. All right. For those latter two parts to happen, you need receptors on your own host cells so the complement can bind and then allow other cells to do its job. These are very important to know. So receptors, you have complement receptor 1, complement receptor 2, 3, 4, complement receptor IG, C5A receptor, C3A receptor. They all have functions, but then there's some tricky points here. So that I just want to draw your attention to, right? CR2 is also known as CD21, right? We find it on B cells. It's exploited by EBV. So CD21, CR2, EBV, hopefully are ingrained in your mind together. Another one that's important is CR3 and CR4 both have something called CD18. What's CD18? It's an adhesion molecule. It's the adhesion molecule that's lost in LAD1. So in LAD1, right, your cells cannot adhere to the cell wall and get to the site of infection, but you're also losing two complement receptors, and two complement receptors that stimulate phagocytosis. There's two mechanisms there. The LAD shuts down your innate immune system and adaptive immune system. So your cells can't get to the site of infection, can't adhere to the cell wall and get out, and you also have lost these complement receptors that would be stimulating phagocytosis. All right. Your head for a second. We're going to go over this one more time. Okay, so 3 pathway starts. This is from the Boss text. I actually like how they put these side by side. The alternative pathway, you get spontaneous cleavage of C3, binds to the microbe. It will bind to B, which gets cleaved by factor D. So then you have C3BBB, which is a C3 convertase. Classical pathway, you have C1, that's going to bind to your antibody, right, IgM or IgG, and it's going to cleave C4 and C2, and a C4B and 2, right, in your textbook, 2B or 2A. I like to just call it 2B, forget what Janeway says officially correct. Right, C4B, 2B is a C3 convertase, okay? Or something very similar to the C1 complex is MBL in the mass, where the ficalin in the mass. It's still just going to cleave C4 and C2, which can bind together to form a C3 convertase on the outside of the microbe. Okay? Each of these C3 convertases, no matter what it looks like, of course, can then convert C3 into C3A and C3B. So a bunch of C3s are cleaved, but one of them can come around and then bind back to the C3 convertase, and a C3 convertase plus a C3B is a C5 convertase. We've made C5 convertases. So these C3B and C3A are active on their own, or you can make a C3 a C5 convertase and cleave lots of C5, and then C5a can go and act very similar to C3a, or this C5b perpetuates that hole punching. It allows that MAC complex, the membrane attack complex, the MAC attack, to start punching holes in the cell. Okay? The C3 convertase is the same in the lectin and classical pathway. It's different in the alternative pathway. Therefore, the C5 convertase is also different in the alternative pathway, but structurally very, very similar, just like these are structurally very similar to the C1 or the META. Okay? Now, something I hope to pay a lot of attention to in this chapter are all these regulators that can work in different ways. Okay? I think we're going to take a look at some of these. Right. So C1 inhibitor we know, of course, because it's associated with what disease? Its absence is associated with hereditary angioedema. Very good. There's a C4 binding protein, complement receptor 1, factor H, factor I, the K accelerating factor, membrane cofactor. And then there's actually one that's fairly unique. There's one called protectin. The unique one, I think, is here, piperidin. So piperidin actually stabilizes the C3b-BB complex. So it is one regulator that is a pro-regulator. So it actually helps its complement cascade perpetuate. So piperidin, which they also call factor P here, but I like piperidin, I think that's what I've typically heard it as, stabilizes this complex and lets this convertase happen. So there are things like factor I. So most of the rest are found on host cells and are going to prevent the complement cascade from attacking a host cell. It's very important. We don't want complement to attack our host cell. So there's something called factor I that can actually inactivate C3b. So it cleaves it and it makes it IC3b, or inactivated form of C3b. Complement receptor 1 apparently can help release this and leaves it inactivated form bound to the surface. Right, so complement receptor 1, there is factor H, MCP, DAF, can all bind to C3P. It can displace BB. So these are all kind of negative regulators. They prevent activation on a host cell where you don't want it. And then factor I, of course, can inactivate. So, right, different stages at which complement activity is regulated. So in the classic pathway, right, you have C1, and you have something called C1 inhibitor, and as the name would imply, C1 inhibitor prevents C1 from cleaving C4 and C2. Okay, so if you have no C1 inhibitor, what's going to happen to C4 and C2? Well, it's going to be consumed. If you're not inhibiting C1, C1's going to keep working, and it's going to cleave lots of C4. So if you were to check your serum level of C4, what are you going to find? No. You're going to find that's low. And we exploit that when we're trying to diagnose hereditary angioedema or acquired angioedema. Something's wrong with the C1 inhibitor, so your C4 levels can be very low. So C1 inhibitor, like Dr. Portnoy very aptly mentioned, all these things have many jobs. Well, the C1 inhibitor also prevents kallikrein from degrading into bradykinin, which can attract fluid and lead to angioedema. Right. So if this is gone, it actually has, I think, three different mechanisms. I think it actually also controls the mast. One of them, right, is to stop C1, which would stop it from consuming C4. One of them is to stop the production of bradykinin. So if it's not there, your C4 is going to be low. You're going to make lots of bradykinin and draw fluid in. You can diagnose by looking at the C4 level, and then, right, what happens once that complement system starts, or once that kallikrein system starts, you're going to make lots of bradykinin, and you're going to have these episodes of angioedema. Right, so another one is at the C3 convertase stage. And this can be stopped by all these things that we talked about before. So like DAF, CR1, factor I can inactivate and displace. And then there's one called CD59 that can actually prevent this C9 from, this poly-C9 from forming a hole and punching in. So once the C5b67 has come in, CD59 can prevent this final step from occurring. Now, there are a couple of diseases that are mentioned that I want to bring to your attention. One being if, I don't think there's a nice table in here like there was in a BOS. But if you have a deficiency in DAF or CD59, so in some ways your complement system is going to be unchecked on, or unchecked on host cells. You may have something called paroxysmal nocturnal hemoglobinuria. Excuse me. If you have mutations in MCP, factor I, or factor H, you have a predisposition to atypical HUS, hemolytic uremic syndrome.
Does that mean the predisposition means if you happen to catch a bacteria that's triggered, or the atypical means just randomly occurring HUS?
I don't think the atypical refers to that. Well, I guess depending on the mutation, I think it's inevitable because you're inevitably going to have microbial exposure, right? Yeah, I don't know why it's called atypical, but I don't think it refers to when it happens. I think it refers more to the presentation of once it's happening. So, especially for second years, takeaway from this chapter is to, have a, I think it's easier once you kind of read about the complement system over and over to know what these proteins do. Very important to get a hold on if you have a deficiency in one, what's the clinical presentation of that going to be. And I think that's a very important takeaway point, which is maybe underemphasized in the Janeway text compared to a BOS text. One of the older BOS, I don't think it's still on there in the newer version, but one of the older ones has a nice table. I'll look for that. Again, that lays it out very nicely, but a really good table to kind of put in your binder of things to study, memorize for the boards.